Good morning. Thank you all for joining and thank you for the opportunity to present. I'm really happy to show you the work that I'm now wrapping up in the group of Christoph Müller and Bo Heinsberg. So let me start with a very big picture of something that everyone is very familiar with in here, so the central dogma of molecular biology. So as you all know, in order to express information that is contained in DNA, it first needs to be transcribed into mRNA, which is done by Pol2, and this is something that we normally all think about. And then it needs to be translated using a ribosome into a protein. And this is something that everyone is completely familiar with, and I'm sure that there are no surprises. But then there are two more very important players that make this whole process go. So we also have Pol1, which makes the ribosomal RNA, which constitutes the ribosome. So without that, the translation wouldn't occur. And we also have Pol3 that makes a lot of short non-translated RNAs, including tRNAs, and these are also obviously important for translation to occur. And while all of you mostly think about Pol2, because it's obviously the polymerase that makes the largest repertoire of RNAs, those RNAs constitute actually the minority of the entire pool of RNAs in the cell. And I will try and convince you that Pol1, which makes ribosomal RNA, which makes up the vast majority of the RNA in the cell, is very important for the functioning of the cell. So RNA synthesis occurs in the nucleolus, and there needs to be a very precise balance of activity of Pol1. So if you get too much of Pol1, you get cancer. So Pol1 is required for cancer progression, and because of that, is also a target for anti-cancer drugs. But then if you get too little of Pol1, especially during development, you get neurodevelopmental disorders such as Treacher-Collins syndrome, which I'll touch upon a bit later. Traditionally, polymerases have been studied in a model organism which is yeast, Saccharomyces cerevisiae, and it's been a great model because it's cheap, it's easy to grow, we can easily draw a lot of conclusions from this model organism. But I don't think you have to be convinced that if you want to study something like cancer or like neurodevelopmental disorders, well, yeast don't really suffer from that being unicellular. So we really wanted to get insights into the human system to be able to draw conclusions that are maybe relevant to the human health. So just to introduce the polymerases, so I'm very happy to say that this is now a gallery of mammalian polymerases, which have been recently complemented by our group and many others. So all of the polymerases are multi-subunit complexes. They share this homologous core, which looks like a crab claw, and it can close on the DNA in the process of transcription. And then on top, you have this protruding subunit, which is called the stalk. Then Pol2 is actually the smallest out of the three polymerases, because Pol1 and Pol3 integrated stably some subunits that are homologous to the general transcription factors of Pol2. So here on the bottom, we call it a heterodimer. This is two subunits that are homologous to TF2E, TF2F, so they function in initiation. And then there are also some subunits that are shared between all three polymerases. They're usually quite small. There are 5 of them, and then additionally Pol1 and Pol3 share 2 more subunits specifically, which are over here, and this is the Biogenesis Platform. So when I approached this project, there was really no system to purify or to study a human Pol1 and Pol3. So I attempted first to tag them endogenously, and I think that this is my link to the cellular section of this conference. So I used CRISPR-Cas, I inserted an endogenous tag, which is a fluorescent M-cherry, and then some affinity tags into the locus of a subunit that is shared between Pol1 and Pol3. I also used this antibiotic resistance gene, blasticidin, preceded by self-cleaving peptide, to increase the recombination rate and to increase the selection so I could easier get a homozygous clone. So then we confirmed that indeed there's no change in behavior of Pol1, so it still localizes to the nucleolus. You can see here the endogenous signal from M-cherry. I did the whole CRISPRing and adherent hex cells because it turns out that the suspensions ones don't really like to be expanded as single clones, so they just die. But then I managed to adapt them to grow in suspension in this special thermal Fisher medium that is called XP, and they grow to very, very high density. So that gave me enough cell mass to actually purify something reasonable. And as you can see on this C stained gel, I have a full complex which is very clean. The yields are not amazing, I have to admit. They're about like 50 micrograms, but this is more than enough for one freezing session of Cryo-EM. So this is the great advantage that we have over crystallography. And I will now cut a very, very long story short because I don't want to talk about the technical details, but you can obviously ask me about that. So after about a year and a half, with a lot of optimization on the grid level and then in the processing, we get a nice structure where we could assign all 13 subunits belonging to POL1 at 2.7 Armstrong overall resolution, and the core it goes to 2.5, so we were quite happy to see that. And I would like to highlight some interesting features that some features and biological conclusions that we can draw from the structure. So first of all, I would like you to focus on this part, which is the RNA exit tunnel. So here is the active site, and in the exit tunnel, RNA protrudes. So once we looked into this channel and assigned all of the density here marked in red that we believed was coming from RNA, we could see that there's definitely significantly more density that would just belong to one primer. So I made a scaffold that mimics the elongation bubble. So it's 2 strands of a DNA, which are artificially open, and then the primer is annealed. And this is something that we have to admit was an honest mistake. The primer is self-complementary at its 5' end. We used the same primer also for preparation of the human POL3, and we never saw anything like this. And only in POL1, we saw this very clear double-stranded RNA helix, which fitted really well into density once refined. So we asked ourselves, why might that be, and what advantage could it bring? So in a work done by the Tollerby lab, they showed that having structured RNA in the RNA exit tunnel might help with transcription and might increase the elongation rate. So the mechanism of transcription is a constant equilibrium between going forward and backtracking for each polymerase. And having this structured RNA in the tunnel might just stop backtracking because it would have to unwind it and then only push it back to the tunnel. So this is the idea why it would go a bit faster and be a bit more processive. And for POL1, obviously, which needs to make very, very long transcripts, it's quite important. Then we also looked at the RLA exit tunnel, and we could see that the channel is much, much wider, about 10 Armstrong wider in the case of POL1 compared to POL2 and POL3. And that would also maybe help fit the RNA and really let it fold inside the tunnel. Another feature that I would like to highlight is the stalk. So this is this protruding part on top of the core. And this was something that was very puzzling when I started the project. And this was also one of the reasons why we decided to use CRISPR. In yeast, there's this small subunit, A14, here marked in pink. And the homologue of this was not identified in humans when I started. And now having the structure, we can see that there simply isn't one. So human POL1 has one subunit less. And it's then 13 subunit complex, not 14, as has been claimed. We then looked into why it might be, and looking here, you can see this N-terminal helix over here that exactly occupies the space which the small subunit would occupy, so this kind of complements it. And the same N-terminal helix is in yeast bound to the core by these two yeast-specific extensions that then don't let it flip out. So this is clearly the difference between human and yeast. And then we asked ourselves, okay, so is it yeast that is weird, or is it humans that are weird? And we looked into the phylogenomics with a collaboration with the Bateman group in BoliVI. So if you look at this in dark pink, this is the small stalk subunit in POL2, and you can see that it's present in all of the different species that we looked at. But then if you look at the small subunit from POL1, this is light pink, you can see that it's only found in some species of fungi. So indeed, we have just chosen the wrong model organism in this case, and it's really important to study the human complex. And this is even more important because we know that this is the platform where a lot of initiation factors bind. So I also saw the structure with initiation factor RRN3, which is POL1 specific initiation factor, bound to the core. So it sits as expected here snugly next to the stalk. And it also explains why it might be beneficial for human POL1 to have a bit more flexible stalk. So the bottom of the stalk here is tightly bound to the core, but then the upper part can swing away by about 5 Armstrong, which we think could be sort of a conformational selection. And this hinge that is here created that allows it to swing away is exactly where this small subunit would sit. So obviously yeast cannot really swing like this. And to further show that this is really an important interaction surface, so it's also a site of known phosphorylations. So if these two residues are phosphorylated, then RN3 cannot bind to the stalk, and this is also a way to control and modulate the function of POL1. Last but not least, going back to the biomedical relevance, given that we have quite high resolution at 2.7 Angstrom, we're quite confident looking at really residues and mutations, especially given that a lot of them are in the core, so we could really see the side chains. So as one would think, there's quite a few residues, mutations that cluster around here, the active site. So obviously, if you have a mutation over there, that might affect the nucleotide addition cycle, so it's not that great for a complex. But then even more mutations cluster over here, so away from the active site. But then the two subunits, RTAC1 and RTAC2, that are shared between POL1 and POL3. So these two subunits are thought to be the Biogenesis Platform, so they would help in assembly. However, the full biogenesis pathway of polymerase is not yet known. And then there are two diseases that are related to the mutations in POL1 and POL3. So Treacher-Collins syndrome and hypomyelic leukodystrophy. So they are both neurodevelopmental. Treacher-Collins syndrome has a lot of craniofacial development issues, patients with Treacher-Collins syndrome, and HLD results in issues with white matter of the brain. So both of them are have a wide spectrum of representation. And looking at our structure, we think that they might affect the mutations related to the two diseases might affect POL1 and POL3 differently, depending where the mutation is located. So I'll give you two examples. So once we zoom into this mutation, arginine 279, in POL1, it makes a lot of contacts, and it's very well shielded from the surface by this POL1 specific subunit. So there's this linker that runs along the edge, and it really shields it from solution. In POL3, this subunit is quite exposed to solvent, so presumably if you have a mutation in here, it will be much worse for POL1 than for POL3. The opposite would be true in case of the N-terminus of the RTAC1 subunit. So the sample is prepared from the same purification, and they are really quite in similar conditions, so I don't think the issue is really from the sample preparation, it's really something inherent to the complex. So the N-terminus in POL3 is fully folded over here, and in POL1 it's exactly fully disordered. And there are at least 4 mutations here that are marked in cyan that are related to the disease. And then obviously they would probably affect POL3 a bit more than POL1. So this is an ongoing work that I'm doing right now, so I'm trying to study this further. So we're doing more and more tests to see if our theory really holds, and if there are really certain differences between POL1 and POL3 in terms of the mutations, and we would like to see if this could have some sort of biomedical relevance, so that at least we could give better prognosis to the patients. However, this is very early stages of the work. And with that, I would like to thank especially my group. So Christoph is a great supervisor who has given me a lot of freedom to pursue this project. Matthias Gierbig has been a PhD student who now has moved on and graduated. We did a lot of work together. He solved the structure of human POL3. Then Helga has done a lot to help me with a lot of cell culture work, and Florence is doing the transcription assays for us. I also have to, of course, point out the amazing Cryo-EM facility at EMBL, because without the maintenance of the microscopes, we wouldn't be able to do anything. Phylogenomics was done by Aleix in Bateman Group, and also with CRISPR, I got some support from Francois and Matteo. And thank you for your attention. I'm very happy to take any questions.